Thank you, Hugues. Thank you for being here. It's a great pleasure to talk at the Collège de France. It's also very intimidating. I wanted to start with the acknowledgements because this is largely work in progress. So a lot of what I'm going to show has been done by Tsukushi Kamiya, who's an FRM postdoctoral fellow in the team, and also Nicolas Tessandier, also a postdoctoral fellow in CNRS, did some of the immunology work. Jacques Ravel from University of Maryland shared some essential insights and also some data that I'm going to present. And finally, the big part of the data I will present, and I'll acknowledge it more specifically, is from the PAP-Clear clinical study that we ran in Montpellier. So this year's theme of the Journée François Jacob really echoes that of our own research, so the social life of microbes. Because in the team, we work at trying to reconcile somehow Pasteur and Darwin, studying the ecology and evolution of human microbes. Sociality was a big puzzle to Darwin. And there's this famous quote, at least among evolutionary biologists, where he says that he was actually scared by the difficulty to explain in social insect communities why you would have neuters or sterile females. And this was really a challenge for his theory, so the sociality in itself. And as I put as a title, why do individuals would go out of their way to help or harm others? Some answers came later. First, a formalization of what a social interaction is. So for an evolutionary biologist, we'll say that an interaction or a behavior is social is if it has fitness consequences for the individual that performs the action and for another individual, which is the most important part. So to try and explain these extraordinary phenotypes somehow, theory came from Bill Hamilton, who came up with this very simple formula or intuition, which is that the cost that you pay at helping someone else can be selected if this cost is lower than the sum of the benefits you provide to others weighted by your relatedness to others. So this way we kind of formalize the evolution of sociality and altruism in particular. Quoting François Jacob, François Jacob usually uses the word sociality in the humanities sense, or sociological if you want, but there is at least one instance where he refers to social behavior to talk about what happens between molecules and cells. And he also figures that if this social behavior breaks, this is how it can lead to cancers, for instance, if a cell is no longer a member of the community. It's quite striking because this idea to use the microscopic level to investigate social behavior came somehow later on. There were many experiments from the end of the 70s and early 80s. This is one of the oldest I know, by Lin Chao and Bruce Levin. What they showed, they used E. coli, which has been presented before, and looked at the capacity of E. coli to produce bacteriocins, so molecules that will kill other bacteria. And what they showed, and this echoes some of the presentations we had this morning, is that depending on the substrate in which they live, so an agar plate or a well-mixed environment, you see that this affects the ability for the cholecinogenic, so the bacteria that produce the bacteriocins to invade. In this well-structured medium, you can get this evolution. And intuitively, it's coming back to the relatedness. It's because in this medium, you have a stronger viscosity of the environment and more relatedness. So answers to sociality from microbes. And this is just to show that, I mean, there's a review from 2001 already on the topic. Many systems, some that we saw today, Dictyostelium, Myxococcus xanthus that we will see this afternoon, phages, and this is a siderophore. So overall, we now have also a body of theory that captures these social interactions, and especially bacteria can compete for resources, they can spite each other, and they can produce public goods. After this long introduction, I'll move to the heart of the topic of what I wanted to talk about today, which could also be formulated as what makes us humans. And on this graph, I'm sorry, it's from a Frontiers journal, but you see the vaginal pH in a lot of mammal species. And what is particularly striking on this graph is this dot here, so humans with non-BV. BV is bacterial vaginosis, I'll come back to that. But humans stand out among all the other species, especially the other primates. We've known this for a while, but more recent genomics evidence allowed us to better understand what is happening. So this is data from Jacques Ravel, who's one of the experts on the topic. When you screen women in the general population, you will find that the vaginal microbiota community is structured very strongly in 5 main categories. The most frequent one somehow is the Lactobacillus crispatus dominated community, and so this shows the frequency of the bacteria. In red, it's Lactobacillus crispatus. So you see this clustering here with almost in some women more than 90% of bacteria that are Lactobacillus. Then we have 2 other CSTs that are somehow similar functionally with other lactobacilli. And so these lactobacilli are what explains the low pH. They produce lactic acid. Then we have Lactobacillus iners. Oh, I'm sorry, it should be CST3, which is kind of intermediate. And finally, the last community, CST4, so CST for community state types, is very different. It's much more diverse, and the pH is higher. And it's kind of the opposite of the gut microbiota, which was mentioned this morning. So in the gut microbiota, the healthy microbiota will be very diverse, and the less diverse one will be dysbiotic. Here, it's kind of the other way around. Throughout women's lives, this composition can change, and it can also change on a finer time scale. This is, again, data from Jacques Ravel's lab, showing 3 women and the temporal dynamics in these women. So in some, you have very stable dynamics. This woman has a vaginal microbiota with Lactobacillus crispatus consistently. This woman is more dominated by Lactobacillus iners, although you see that there are periods of increased diversity, and these, in fact, correspond to menses, which are shown here. So we know that menses are going to destabilize the bacterial community. And finally, in other women, you have an extremely diverse profile that persists. And this one, there's a predominance of Prevotella and Atopobium, which is now called Fannyhacia. So different types of dynamics. Thanks to microbiologists, we also know much better what's happening at the molecular level. And this is a schematic from a very recent review on the topic. So Lactobacillus crispatus, and also Gasseri and Jensenii, if you want the good or optimal microbiota, will feed on the glycogen that is freed by the epithelial cells. And they will produce bacteriocins. They will also produce lactic acid, which decreases the pH. So altogether, this acts to limit the growth of non-optimal bacteria or pathogens. On the other hand, the suboptimal or non-optimal microbiota, very diverse, often dominated by Gardnerella or Prevotella or Fannyhacia, these can also feed on the glycogen, but they will also feed more or less on the vaginal mucus. And they do this because they can produce enzymes, sialidase, that can cleave the sialic acids at the top of the sugar chains of the mucus, and then eat up the whole fucose, so the sugars of the mucus. In the end, this can end up in also lysing the cells. There's also biofilms being formed. I won't go into this, but what is important is that these bacteria can use glycogen, but also secure an additional resource. They also produce biogenic amines, which are compounds that can somehow counter the effect of lactic acid. So altogether, what I wanted to show with this slide is that we have a fairly good understanding of what's happening at the molecular level. What we wanted to do in the team, and that was Tsukushi Kamiya's work, was to try and formalize this with a mathematical model. And we used the community ecology approach. This is not the first model on the vaginal microbiota, even though there are very few of these models. But most of the models so far have really only focused on the bacteria communities, using what we call Lotka-Volterra models, seeing what's the interaction between one bacterial species and another. Here with Tsukushi, we also wanted to add the resources into the play. So we have two types of bacteria or communities. One would correspond to a lactobacillus, so optimal community, and one to a non-optimal, so let's say Gardnerella vaginalis dominated community. Both of them can replicate. Both can use glycogen, as I said before. You have these arrows here that contribute to replication. But the difference is that Gardnerella can use these sialic acids or private goods, if you want. And these private goods are also released actively through these cleaving mechanisms here. So you get this external loop for Gardnerella vaginalis. The important pieces of the model are also the antagonism from Crispatus towards Gardnerella. This captures mainly the pH effect, but it can also capture additional processes like the bacteriocin production. And here you'll also notice a self-regulation loop. It's because at some point the pH becomes detrimental also for Lactobacillus crispatus, so there's kind of a self-inhibition. This is captured with the system of ordinary differential equations. It's already fairly detailed, but I want to stress that we made an effort to really keep it as simple as possible. There are lots of ways we could extend this model, but we wanted to work with like a minimal version of these two resources and two species. Jumping to the results, this is what we observe, the main feature, if you want, of the results. On the x-axis, what we vary here is the proportion of the resource supply that is private to CST4. So it's the proportion of resources that corresponds to RP. The more you go into that direction, if you want, the more the resources available come from the mucus and the sialic acid, and the less glycogen there is. On the y-axis, it's the proportion of the bacteria that correspond to CST4. So if you're on top there, you have a non-optimal or dysbiotic state, and if you're here, you have 100% lactobacillus dominated. I should stress that in the model, we assume that resources equilibrate really rapidly. Here, what we're looking at is also kind of a rapid equilibrium of the bacteria. So if we start from a lactobacillus-rich environment, you see that the more you increase the private resource supply to CST4, the more the proportion of CST4 increases in the population, which makes sense. They have this resource that lactobacilli cannot access. But there's a striking behavior that occurs right after this 0.5 threshold here, which is that you will jump directly from an intermediate state with both types of bacteria to a state entirely dominated by CST4. And the second striking feature is that if you start from this non-optimal state, so you want to somehow correct it, reducing or putting more glycogen in a way, you see that in order to restore the lactobacillus-rich environment, you need to go beyond the threshold that you passed and where things went bad. You need to go all the way up here to restore the lactobacillus. This is a phenomenon that is known in physics as hysteresis, and we were particularly enthusiastic to see this kind of behavior in the model because it somehow captures what clinicians will tell, which is that it's very difficult to restore a CST4-dominated bacteria, so a Gardnerella dominated, back to lactobacillus. People have tried probiotics, people have tried antibiotics. It's basically easier to go that way than to go that way. Of course, this is just a mathematical model. We did some sensitivity analysis that I'm not going to show, but we wanted to test this with actual data, and this is where Jacques Ravel could be helpful, because he ran this study in the US, mostly involving women suffering from vaginal dysbiosis, so bacterial vaginosis, which is a form of dysbiosis. These women were asked to self-sample twice a week. And overall, here we could analyze 437 samples from 56 women, samples for which we had information both about the microbiota and the metabolomics. So for the microbiota, we used 16S RNA DNA sequencing, so metabarcoding, to identify the proportion of the lactobacillus or Gardnerella. And for the metabolomics, we used information related to glycogen, so glucose, maltose, maltotriose, and sialic acid to identify the proportion of resource. I want to stress that one of the advantages of having an underlying mechanistic model is that we're not directly just looking at the proportion of sialic acid, which would be problematic because sialic acid is used by the bacteria but also released by CST4. So it's kind of a flow. And this flow we can express from our equations. And this is what I show here. You'll see the proportion of Gardnerella vaginalis bacteria, the proportion of sialic acid in the resource, the total concentration of bacteria, total concentration of resource, so things that we can get from the data, and additional data we threw in. Lambda 4 is the difference in growth rate between Gardnerella and Lactobacillus. So again, what we're looking at is really a flow. And going straight to the results, Tsukushi structured the data in terms of the quantile of proportion of resource supply private to CST4. And what you see is that in the first quantile, so women who basically have less sialic acid, we rarely see CST4. We see a majority of CST1, 2, and 5, and a lot of CST3. And then the more this resource increases, the more you see the predominance of CST4. So I think that's what's written here. The proportion of sialic acid is associated with the increase in CST4. So the data kind of supports the insights from the model. Another actor that I wanted to mention briefly is also the immune response. It was evoked this morning. In this community, in the vaginal environment, you have the bacteria, but they are also interacting with the immune response. And what this slide is meant to say is that basically we know little about the immunity, the local immunity in the female reproductive tract. And there's this question as to whether the immune system is shaped by the vaginal microbiota or the other way around, or both. And we wanted to investigate this with some data we collected that I'll present later in details, but I just want to show you what it looks like. I mean, we know we expect to find these kind of cells, resident macrophages, perhaps Langerhans cells, perhaps TCR gamma delta, but it was poorly characterized. And Nicolas Tessandier was able to sort some of the data we collected, which was a nightmare, because for those of you who do flow cytometry on a cervical smear, you don't want to do that. And by using automatic gating, he was able to identify relevant populations. So you see the usual suspects, CD4 T cells, CD8 T cells, but he also showed that we have a lot of neutrophils, some NK cells, and quite some TCR gamma delta cells. Even though, I mean, this has to be also put in with respect to the markers that we used. We were targeting these kind of cells. What is interesting is that when you compare CSTs, you can try and compare these immunological profiles. And Nicolas compared the CST3, so the one that is in between optimal and non-optimal, with CST1, which is optimal. And you see that there's kind of an enrichment in CST3 and neutrophils, a slight signal for monocytes, and here a decrease in the TCR gamma delta from a mucosal origin. The signal is even stronger when you compare the really non-optimal community, CST4, with CST1. So here you also see the CD4 T cells and CD8 T cells that are increased, and this is consistent with existing data, which show that in bacterial vaginosis you have higher inflammation, and it also explains why I mean, it's one of the hypotheses as to why women who are in CST4 are also at higher risk to be to contract HIV upon exposition. And you see the signal for TCR gamma delta cells from mucosal origin that are rarer, that is even stronger. So it's ongoing work, but it's a contribution to show that the immunity seems to be shaped by the type of vaginal community. And finally, so we talked about the social lives of microbes at the within host level. how they deal with the immune response. One of the interesting things of microbiota is that you have multiple levels of adaptation, as Lewontin formulated it, which is that the fitness, if you want, can be measured within the host, but also at the between host level. And I wanted to mention this in also the context of Canguilhem, French philosopher who, it's more of a French philosopher, but medical doctors all around the world are more familiar with it. He really changed the view of how you define the normal and the pathological. And the vaginal microbiota is a perfect system to think about this. First, because there are associations between the vaginal microbiota and diseases. We know that women who are in this CST4, so the one that is diverse and with few lactobacilli, are at higher risk of contracting STIs like HPV or HIV. There's an increased risk of in vitro fertilization failure. There's an increased risk of preterm birth. There's perhaps increased risk of UTIs. It's more debated, and there are also pelvic inflammatory disease. Where it echoes Canguilhem is that, and I've shown this before, these CSTs vary through time, so you can't just define a woman through her CST. It's a more dynamical perspective. And also related to Canguilhem, one of his views of disease is that health is basically how fast you can recover from a non-optimal state. And again, with the vaginal microbiota, we have these transitions between non-optimal and optimal. And so this is what we wanted to tackle here, this switch between non-optimal and optimal. How do you switch between, how do women switch between these communities? For this, we used data we generated in Montpellier for a somehow different topic. It was to follow HPV infections. But to make a long story short, women were followed every 8 weeks. And we collected samples at the clinic every 8 weeks. And they also self-sample at home. Analyzing the data, Tsukushi was able to extract the data from 125 women who had at least 3 samples. The median duration of follow-up was 20 months, and we generated 16S data for 2,000-ish samples, 500 on site and 1,500 at home. And perhaps the main challenge was the amount of covariates we had to explain this data. We knew everything about these women. They were filling questionnaires every week. And that was kind of the challenge there. This is what the raw data looks like. So here, each line is one of the participants. This is the follow-up time. And the CSTs are colored in a bluish for the optimal CSTs and yellow or red for the suboptimal or non-optimal. So you see that some women are fairly consistent over time, and others switch a lot between CSTs. This, again, is the raw data. You see here, this is the number of transitions we observe, so from 1 to 1, from 1 to 2, from 1 to 3. If you look at the diagonal, so the women who remain in the same CST, you see that we have a lot of CST1, which is consistent with what Jacques Ravel has in terms of epidemiology, and then CST3, but also a lot of the CST4. I want to stress that this is one of the first data of its kind for France, I'm sure, but even for Europe. Most of what we know about the vaginal microbiota comes from North America and a little bit from studies in Africa. Okay, so the goal here was to try and understand these switches, but not just getting probability of switching, which is easy, but trying to find if covariates are associated with that. This was particularly challenging given the number of covariates. And I'll really skim through the pipeline. I mean, this is also ongoing work, so feel free to ask me questions at the end. But Tsukushi worked in 4 steps. The first one was to clean the data, so to remove variables that were too skewed. Say, if 95% of the women have the same behavior, this behavior won't be very informative. The ones that have too many missing points, the ones that are too correlated between each other, I mean, cleaning the data. He enriched the data by imputing missing values. And then he fitted a Markov model. So Markov model will tell you these probabilities to switch between states. And he used the Bayesian approach, which is quite recent, to try and maximize the number of covariates he could put in the model. To give you an intuition, the idea is that in the Bayesian framework, the prior he puts is really centered on 0. So basically, it will return a 0 effect unless there is really a lot of signal, which is, I think, what is written here, the shrinkage prior. And then he made the inference for the combined output. this is what the data is looking so far. Given the proportion of communities, he could only look at switches between 1 and 3, 1 and 4, and 3 and 4. And he also had to make the assumption that something that will lead you to switch from 1 to 3, it will be the opposite effect for switching from 3 to 1. So, by the way, here on these lines, I don't know if you can read, but you get a kind of an idea of the diversity of covariates we have, from using intimate wipes to using tampons to having a new partner. To show you a little bit of some of the insights we're getting, so focusing on the strongest effects. For the switch from 1 to 3, one of the very strong effects was alcohol. It's interesting in two ways. The first is that our alcohol variable is structured, so we asked them if they didn't drink, if they drank less than once per month, etc., and every day. And you see that the effect, which is a hazard ratio on the transition probability, increases with this intensity of alcohol consumption. And this echoes, I mean, you can find anything in the literature, but there are studies on link between alcohol consumption and bacterial vaginosis. For the switch from 3 to 4, what you see is that condoms and using intimate wash soap are protective of switching from 3 to 4. So we could ask for funding from condom manufacturers and soap manufacturers. Perhaps more surprisingly, we found that being infected by a high-risk HPV increased this probability to switch to a CST4. This is what is shown here. And it echoes some of the work in the literature. People have looked at these associations between vaginal microbiota and HPV, so it definitely warrants further investigations. I need to thank the study participants who were extremely involved in this study, and the data I showed would not have happened without them. It would not have happened also with a lot of help from advisors who helped set up the cohort and the analysis. And Tsukushi and Nicolas were particularly involved for this particular aspect of the analysis. So in conclusion, I hope I've convinced you that the vaginal microbiota is an ideal study system to study interaction between humans and their microbes. But at the same time, it's also an interesting study system on a social side, if you want to stick with a social life metaphor, because you're tackling important public health issues like sexually transmitted infections, fertility, or more generally women's health. There are numerous paths that we're exploring right now, from community ecology to genomics, immunology, epidemiology, and finally evolution, because as I started, this specificity of the vaginal microbiota in human as an evolutionary biologist is one of the most fascinating questions. And with this, I'll thank you for your attention and be happy to answer questions.